So today we're talking all about the microbiome, but not in humans. But to really get started on this, we have to start with the human microbiome. And this is a topic that I find super fascinating because as humans, we are not alone. We contain multitudes. And this discovery of the human microbiome, or the other phrase for the compendium of all the microbes that live on us and in us and ourselves, really altered our understanding of our relationship to microbes. They went from being these tiny critters that cause disease to this really nuanced relationship that enables us to be healthy, but can sometimes turn on us. And some of the cool findings were that there are 10 times as many microbial cells as human cells in the human body, and that all of these organisms that make up the human microbiome contain 46 million genes in just the mouth and the gut samples combined. And this has led them to predict that if you were able to tabulate all of the genes in the human microbiome from every nook and cranny where these microbes are, that there would actually be more genes than there are stars in the sky. And that is on the order of several trillion. And GenSpace, I think, really loves the human microbiome and microbiome science. So what are some of the benefits of the human microbiome? What do they do for us, and why do we like them so much? Does anyone want to share some of the positive relationships that we have with the bacteria and archaea that we cart around with us?
You have a response in the chat from Sibel, training our immune system.
It's also in direct
yeah.
connection with the brain as well.
Yeah. Oh, yeah. So first, these our microbiome helps us to digest our food. They make vitamins that we can't make ourselves, like vitamin B12. They also can, in a sense, control the way we think. Vitamin K, vitamin B12. They can control the way that we think, as I just said. They are thought to have some link to autism and other conditions like that. So really every aspect of our physiology is sort of comes down to maintaining a healthy relationship with our microbiome, from the way that we think to the way that we digest our food, to even body odor. And that's caused by an imbalance of particular microbes on our skin. So lots of benefits to the human microbiome.
You have comments in the chat. They produce metabolites that work as neurotransmitters and hormones, so they talk to our body, from Sibel. And Marge adds that our native microbiomes outcompete potential disease causing microbes.
Oh yeah, those are great. Those are great things to add. So the fact that we have all of these healthy microbes that live in our gut means that it's much harder for pathogens to find a way in and cause disease, because they have so many more healthy, friendly microbes to compete with. That's a really good one. And then what was the other one? Yeah, they produce metabolites and neurotransmitters, hormones, things that help us to regulate our own physiology. So we really, we could not do this without them. We could not live healthy lives without our microbiome. And this is Desiree says, a foreign microbe in the gut. So if anyone has heard of, sometimes by either drinking kombucha or doing more extreme therapies like fecal transplants, you can reprogram your microbiome to enable you to live more healthily and reinstate that healthy relationship with the critters that live on us and in us. But the microbiome isn't just restricted to humans. The microbiome is universal. Does anyone know who this cute little critter I have on the screen is?
A squid?
It's a squid.
Octopus?
It's a squid, and this is called the bobtail squid. And it's really, really tiny, a couple of centimeters long. And it is extremely cute, but it's also extremely important for sort of propelling the idea of beneficial relationships between organisms and their microbiome, or the microbes that live within them. So this bobtail squid has this little organ on its head here that you can see on this this image on the left, and it's called a light organ. And these squid are born sterile, but what they do shortly after they're born is they recruit this particular microbe called Vibrio fischeri that you can see growing on a plate here. And this microbe glows in the dark. And the way that they use this microbe in the light organ is that they shine a light down, and they do this at night. And so they don't cast as much of a shadow. And that means that their predators can't see them as easily, and then they don't get eaten. So they're using this microbiome as a way to avoid predation. So that's a pretty extreme way that an animal can use a microbiome in the ocean. So not just limited to humans, animals as well. And then coral reef are also organisms that rely in the ocean that rely on a microbiome. So coral reefs, beautiful, vibrant, colorful organisms. But if you were to zoom in on a coral reef, you would see that the thing that is actually making them colorful is these, you can see here in the micrograph on the top right, all these little green specks are this microscopic phytoplankton that is called zooxanthellae. And these are this association that coral reefs here in the sort of sea anemone looking thing has with these phytoplankton that enables them to survive by making food in the form of photosynthetically fixed carbon for the coral host in exchange for shelter and waste products that they need to grow. And when you hear about coral bleaching or the death of coral reefs in response to climate change, that coral bleaching is the corals suddenly expunging all of the zooxanthellae, their microbiome that they grow with. So the death of the coral reefs are really sort of like a loss of their microbiome that is leading to their demise. So lots of different examples of the microbiome in play in the ocean. Every critter has a microbiome, from turtles to fish to coral reef. But what if you don't have a body? And that's sort of what we're talking about today. If we were to zoom in on this marine system into a droplet of water, we would see that just like the macroscopic view, it's teeming with life. And these are marine microbes that, zoom pun. Yeah, that is probably a Zoom pun that no one has ever said before, a marine microbiology Zoom pun. But if we were to zoom in, we would see not something boring or talking heads, but these wonderfully diverse marine microbes that are too small to see, but critically important for keeping the Earth habitable and these marine ecosystems churning. So these microorganisms in marine systems, they don't have a body per se in the traditional sense of like a multicellular organism, but recent research has shown that they do associate with other microbes. And so could this be them sort of cultivating a microscopic microbiome of their own? And one particular very abundant and very important marine microbe are the diatoms, which you can see here. And as I said before we started the class today, diatoms are truly my all-time favorite microbe. They are these microscopic organisms that are hugely diverse, and they are eukaryotes, so they have membrane-bound organelles, just like we do. But instead of having multicellular bodies, they live in these single-celled glass houses. So it's two sides of a shell that are made out of glass that sort of fit together like a petri dish that you can see here. So in this middle micrograph, you can see them hamburger style, I guess. And each of these little dots is a chloroplast, one of their membrane-bound organelles that enables them to photosynthesize. And then you can see them hot dog style, the same species flipped on its side. So you can see they're sort of like a hat box or a petri dish. And what is really cool about these organisms, other than the fact that they're just stunningly beautiful, is that when they grow and divide, the two daughter cells form within the old cells, and then one daughter cell is just ever so slightly smaller than its parent, and one stays the same size. If you can imagine two new cells forming within the base and the lid of the petri dish. So what happens over time is that one side of the lineage continually gets smaller and smaller, and one lineage stays the same. So that's what we're seeing in this top right micrograph. These are exactly the same species of diatom, but one has stayed the same size. And then there's this itty bitty little one that has continued to get smaller with each division. Tiny little babies. Yes, they're very cute, but you can imagine that if they kept dividing forever, they would ultimately disappear into nothing. So what diatoms do is they have sex. They can reproduce, they can create sperm and egg cells and basically reproduce like much larger organisms do. And so they either will come across each other and reproduce that way, or send their gametes into the water to reproduce. And then when a male type diatom meets a female type diatom, they will make this new larger cell that sort of buds from within the old one, and then they will return back to their normal size. So Desiree asks a question, where do we find diatoms exactly? Everywhere. Any place there's water, be it freshwater or marine, there will be diatoms. When a puddle forms on your Brooklyn sidewalk after a rainstorm, you are more than likely to find diatoms in there. They're so pervasive in aquatic environments that forensic scientists even use them to determine if a victim that they find has been drowned, because they can look to see if there are diatoms in lung tissue as a way to determine cause of death. That's a really horrific example, but it goes to show that diatoms are everywhere. Drinking water too. Even filtered drinking water can contain those little microscopic glass shells. You would have to filter a lot of drinking water in a place like New York City to find them, but they're there. In wine, probably not in wine. Well, I don't want to say never. Biology has an exception to every rule, but you would probably have to filter a lot of bottles, and it would probably be a byproduct, because you don't add water to wine unless it's really, really bad wine, I guess. Anyway, the reason that we care so much about diatoms, though, is because they are hugely important to keeping our planet a habitable place. 1 in every 5th breath of air that we take is oxygen that was produced by a diatom. And so they are critically important in ocean ecosystems for being food for organisms that are higher up the food chain, all the way to whales, to us, and making the oxygen that we breathe. So they're beautiful, and they are, critically important to keeping the Earth a habitable place. There is also a lot of art associated with diatoms. So one of the optional readings we sent along that if you haven't had time to look at, the video is just amazing. But these secretive Victorian artists used to use the very first microscopes to organize microscopic micrographs is the scientific term, but I guess like little greeting cards that they would send to each other and display at their parlor gatherings. And just stunningly beautiful. And if you can imagine that each of these critters is about, 50 micrometers across, that would be very tedious. So for all the more art-leaning people in the crowd, why would this be such a precious gift to a Victorian, or to me as someone who loves diatoms? But particularly the Victorians. Does anyone know when the microscope was invented? It was right around the Victorian time. I'm not so good with the ages, but in the 1700s, I believe, which I think is right at the beginning of the Victorian period, though someone should correct me if I'm wrong. And so this was such a precious gift because it was very rare that you would be able to even see this. Van Leeuwenhoek in the late 1600s. So very few people had microscopes, and very few people have them today, but it was an extra precious thing to be able to receive this sort of secret message that very few people could see. And at that time, they did not even know the massive climatic importance that diatoms had. So if anyone wants to send me one of these as a thanks for leading the class, I would gladly accept it and look at it under my little field microscope that's on my desk. But diatoms are not alone either. So this is an even closer zoomed in electron microscope image of various diatoms. And if you sort of squint and look at some of these, you can see these tiny little rods and cocci that are growing on these diatoms. So these intricate geometric patterns are the diatom's glass shell. And then these little critters that are growing on there are bacteria. And so these types of associations were the first indication that diatoms might glean some benefit from associating with microbes. So why would a phytoplankton want to have a microbiome? What could these two microbes potentially get out of this association? And a hint for thinking about this is to think about how we glean benefits from our microbiome. What do we get from the bacteria that live on us and in us? And how could phytoplankton benefit from that as well? Does anyone have any ideas?
Desiree from the chat says a mutual beneficial relationship. Sibel says nutrients.
Exactly, yeah. So living in the ocean is a super dilute environment. It's very hard to get the nutrients that you need to grow. These diatoms are exactly like plants. They essentially need fertilizer like nitrogen and phosphorus to grow. And the waste products of bacteria could be nitrogen and phosphorus, and so they're getting this mutually beneficial relationship that gives them nutrients. And this could be a way that they can absorb or take up these nutrients and enable them to grow and be more successful. Yeah, that's exactly right. So after people saw these associations, they started looking into the nitty-gritty relationships between what the give and take might be between diatoms and the bacteria or microbes that live on them. And early work showed all different types of relationships, from parasitic to competitive to synergistic, which is just a fancy way of saying you pat my back, I pat yours. So some examples that Desiree and Sibel brought up were exchanging nutrients in sort of a mutually beneficial relationship. So you can see here, this little bacterium is giving ammonium, a waste product, to the diatom. And well, this wasn't a good example because they didn't know at the time that there's a question mark here, but that the diatom is giving something back to the bacteria in exchange for that. Or here, a vitamin B12 in exchange for dissolved organic matter or delicious carbon compounds that these bacteria can eat. So these relationships are sort of strikingly similar to the relationships that we have with bacteria. Human bodies, we can't make B12 ourselves. We rely on bacteria to do that for us. And the exact same thing is happening in these diatoms. So even though they don't have a body, in the traditional sense, it's very likely from this early evidence that they are relying somehow on cultivating this microbiome, even though the organisms that in their microbiome are unicellular, and they are too. And so the final question before we get into this paper is, what are the challenges that these marine microorganisms face when it comes to maintaining a microbiome in this without a body and in the... Anyone have any ideas?
Can you repeat that? You froze.
Oh, sorry. So my question was, considering that the ocean is this liquid environment that has dilute nutrients, what are the challenges to getting and maintaining a microbiome? So I see Marge said attachment. Exactly. Yeah. I have this picture, which I realized I didn't explain and just popped up, of this little kid jumping into a pool of Jello. So, as humans, we can swim relatively easily if you know how to swim through water. But if you're a microbe and you're very small, the physics of existing in a liquid medium completely changes, and it's like being suspended in Jello. So imagine trying to move around in an enormous pool of Jello. It would be super hard to get from one place to another. And then once you get there and you find something like a diatom as a bacterium in this super dilute environment, then you have to attach. And before the dispersion and diffusion takes you away. So all of these challenges that make it so much harder to get and maintain that microbiome in a marine environment when you don't have a body. Yeah, and forming a biofilm could be a way to maintain that microbiome. So sort of having maybe like a sticky sheath around what you want to stick on would enable you to stay for even longer. So I just jotted down some examples, which I think we all talked about. You don't have a body, no body to keep your microbiome inside. Microbes are small. The ocean is super dilute. And for a microbe, it's like swimming through Jello, trying to get to something you want and stay there. So these are all things to keep in mind as we talk about the paper for today's journal club. Before I go on, does anyone have any questions or comments or things that they didn't understand? I'll take this opportunity to remove the many hearts and stars. So many hearts for the squid. Yeah, the bobtail squid, super important for marine microbiome research.
And I'm not sure if you'll go into the, probably not, but also like Vibrio, especially with the bioluminescent aspect of Vibrio, has also helped give a better understanding of how bacterial cells communicate in a community, in terms of, you look it up, something called quorum sensing, and just knowing how many in how many cells are around them, what activity they should be doing. I know it's unrelated to this, but Vibrio, and in particular, I'm pretty sure Vibrio fischeri is a species that has been key in sort of understanding quorum sensing. That's it. That's all I wanted to say about that.
Yeah, no, thanks for bringing that up. That, I think, arguably Vibrio fischeri and bobtail squid are even more important. And Bonnie Basler, who is a key researcher in how bacteria talk to each other, using this chemical signaling called quorum sensing. I think she has a TED talk, or probably a few TED talks, or talks that you can find on YouTube.
Yeah, she has a great TED talk.
But that, I mean, yeah. So the way that the Vibrio fischeri that live in the bobtail squid's light organ can turn on and off is based on quorum sensing, like Marge said, and them essentially talking to each other and figuring out what chemicals they use is sort of like the Rosetta Stone of tapping into how bacteria talk to each other. Super cool. We should talk about that at a future Planet Microbes. But yeah, arguably the more famous thing that bobtail squid are known for. But today, the microbiome and diatoms. So this paper is all about, how diatoms get over these enormous challenges in recruiting and maintaining a microbiome in the ocean when they don't have a body, when it's salty and there are waves knocking you around and it's like swimming through jello. So the first thing that these researchers needed was a model organism to do this study on. So they used this organism called Asterionellopsis glacialis, which I will just call the diatom from now on. But they isolated it from the Persian Gulf, and they did two different things to it. And what you would have seen in the paper is them saying this terminology for xenic and axenic. And this is basically a fancy way of saying with microbes and without microbes. So they treated some of these cultures with antibiotics, just like you would treat yourself if you had strep throat or something. And they got rid of all those microbes and formed the axenic diatom, acclimated it to no longer having a microbiome. And then they also isolated that community of bacteria, or the microbiome, and then grew that separately, which they used to sequence genomes and then to reseed the diatom culture that they had stripped the microbiome off of. Does anyone have questions or stamps on any part of that doesn't make sense, or comments or anything at all? Pretty simple so far. And then we get to the actual part of the experiment. I personally found this graph super complex to look at and figure out what was going on. So basically, they looked at the separate microbiome and first determined who was in that microbiome. And that's this center donut plot here. So you can see basically the percentage breakdown of the different critters that make up the microbiome of the diatom that they isolated. So mostly flavobacteria, and then Rhodobacteriaceae and other. So that's the first part. And then they wanted to figure out what happens to the physiology of the microbiome and the diatom when they are alone, without each other, compared to when they re-inoculate that axenic diatom that was growing without its microbiome with the original microbiome. So they took that clean diatom and they popped a slug of the microbiome back in, and then they looked at the gene expression in the bacteria that were growing in there, as well as the diatom. So these triangle plots basically show how different gene families, which is basically a proxy for different functions, like glycolysis or processing sugar or eating nitrogen, things like that, respiration. And each side of the triangle is how all of these different gene families for those different functions changes when the bacteria are alone versus back with the diatom. So you can see based on how the little dots are moving that in the microbiome, at least, when you are back with the diatom, you are completely reprogramming your physiology. You're doing totally different stuff. And then what I think is super cool is if we go over here to the right panel, we can see how the diatom or the host gene expression changed. So in this sort of, I guess, also a donut diagram, each line here you can see the sort of variegated pattern is a different gene in the diatom's genome. So when it's green, the expression is, I can't remember if it's up or down. Your little Brady Bunch faces are blocking. So if it's green, the expression turned up higher of that gene. And if it's red, the expression turned down. And this outer donut is after just 30 minutes of being reunited with their microbiome, enormous swaths of their genome completely reprogrammed, which is, I mean, it's sort of astounding that within just 30 minutes, they were changing expression of all of these different genes. And then after 24 hours, which is the inner circle, that has completely reprogrammed again. So these genes that had turned on in the first 30 minutes, after 24 hours, those weren't being expressed as much, but this whole different swath of genes had turned on again. And so I think this is just really striking evidence that the microbiome is critically controlling how both diatoms and their microbiome are existing in the ocean. Super cool. They also looked at the metabolites. So the metabolites are all of the stuff that the cells are excreting, what they're spitting out into the media around them. So that would be the things that are directly related to what the host and the microbiome are exchanging. And these PC plots are basically a statistical way to say, is one sample different than another sample? So here on the far left, we can see all of the red samples are all of the metabolites that were excreted by the azenic or no microbiome diatoms. And the green are the reseeded or with microbiome, and the metabolites that they're excreting completely different. So this is evidence that that the changes that they see in the previous diagrams, it's not just genetic. They're actually changing what they're excreting into the environment, which means that this could have profound effects both on, or more evidence that it could have profound effects on the organisms that are there, but also the environment at large. And you can see similar changes over time in the azenic cultures of diatom and the diatoms that were reseeded with that microbiome. So you can see sort of those key differences over time recapitulated. Anyone have questions so far? Alrighty. So then they found these two weird compounds that the diatoms were producing, and they wanted to figure out what these really highly produced compounds would do to the microbiome. And one of these was rosmarinic acid, which I had a mental note to look up, but I forgot until just now, but probably it's called this because it was first isolated from rosemary. But if someone wants to Wikipedia that and confirm or deny, I'd be very curious to know. A lot of these metabolites were named after what they smelled like or where they were found. So that's my best guess here. But in this system, they figured out that rosmarinic acid was able to reduce the motility and increase the adherence in these really key microbiome species for this diatom. So to test this, they took 2 different species of bacteria. Oh boy, these names. S. pseudonychiae and Phaeobacter. So these are the friendly microbiome species that grow really happily with other diatoms. And then they also tested it with this Alteramonas macleoidei. And this is a bacterium that often grows with diatoms, but they are direct competitors for the diatom. So they use the same stuff, and they are not really, helpful members of the microbiome. They're competitors. So they looked at what happened when they added rosmarinic acid to these 3 different bacteria. In the friendly species, they saw that motility was reduced and their attachment increased. So these gray bars in the left graph are when they added rosmarinic acid, the motility went down. And then if you look over here on the right, you can see these are kind of fuzzy graphs. So all of these circles around here aren't anything alive. These are just the pores on the filter. So they were growing these diatoms and bacteria in liquid, and then they had to filter them flat onto essentially a coffee filter so that they could put it on a slide and put it under the microscope. So as all of these itty bitty little green dots are bacteria, and these larger circles are the nucleus of the diatom. So all of the bacteria that are not attached to the diatom would have been swept through this coffee filter, and those that are attached would still be there. So you can see that with rosmarinic acid, you see they saw an increase in the bacteria that were adhering to the diatoms in both these 2 different bacteria. And then when they did this with the unfriendly competitor bacterium, they added rosmarinic acid and they didn't see hardly any attachment. So essentially, this compound is making the microbiome a little bit lazy. If you're not motile, you're going to want to settle down, attach, stick around where you are. So this is evidence that these diatoms are producing this compound to selectively recruit these friendly microbiome species that they want to live around them. They also tested this other compound called azelaic acid, which I would assume was first isolated from an azalea flower, which is nice, probably smells good. And what they found is that when they added this azelaic acid to the friendly species, it enhanced their growth. So the cultures where they added this compound to are in the squares, and you can see they're significantly higher in abundance, which optical density is a proxy for in those cultures. And then in the competitor, the adding azelaic acid caused them to grow less well. So another example of how this diatom is producing these compounds to sort of curate their microbiome. And then finally, the final question, which I think is a super cool part of this study, is this is all well and good. It's happening in a test tube, but are these types of interactions important in the field? And so they figured out that knowing whether certain bacteria are responsive to azelaic acid you can test that by looking for a particular gene that bacteria have called AZER. And this is the azelaic acid transcriptional regulator. And so the presence of that gene will tell you whether or not a particular bacterium is responsive to azelaic acid. If they have it, then most likely when a diatom produces that compound, that bacterium will grow better. And so these researchers took advantage of this enormous global database called the Terra Oceans database, where this little yacht by French researchers in a lab where I did my postdoc went out all around the oceans and collected samples and sequenced all of the DNA in depth profiles from each of these points around the ocean. So it's now this publicly available database where you can query any gene you want and see if it's in those samples all throughout the entire ocean. So they looked for the presence of this azelaic acid transcriptional regulator, and they essentially found it everywhere, but with different species that were responsible for producing it. So these donut plots show in the center bacteria at the surface ocean, and then bacteria at the deep chlorophyll max, or the lowest point where photosynthetic organisms can healthily grow. And they saw it everywhere. So this is great evidence that these processes are actually happening in the field. So at the end of the day, the main result for this is that these diatoms are using these compounds, selectively curating their microbiome. And I think that's super cool. Even if you don't like diatoms, I hope you would think that that was pretty cool too. So if does anyone have any questions before we hop into the discussion questions or comments or anything? Oop, I'm way behind in the chat. Yeah, but really anything at all that you want explained more, or questions or comments, art ideas, applications to our own microbiome. Yeah, exactly. That's one of the how to curate our own microbiome that Desiree just said. That is, there are a ton of startups that are looking specifically at how can we really selectively and specifically make our microbiome tailored for optimal health. You can eat a healthy diet, but if there are these specific compounds that you could take instead, maybe that would be a much quicker way to build a healthy microbiome. Probably not as cheap as kombucha, but anyone else have any other questions or comments?
I think I just wanted to comment a little bit on sort of the time frame when these sort of interactions occur. Like we like to think that these can happen immediately, or these sort of curations happen, oh, it's, I'll squirt a little bit of this rosmarinic acid and you'll come stick to me. But really, when we think about it, it's really evolutionary timescales, which are shorter than ours because microbes, have shorter life. I don't want to say expectancy, but you know what I mean, life cycles.
Yeah.
But these sort of adaptations where you might attach, and it's sort of a trial and error situation over geological timeframes that these happen, and to then be able to translate that to the question about looking into curating your own microbiome. Think about how incredible it is to extrapolate a geological time frame where trial and error and evolution and environmental factors have created this incredible symbiosis, and now to apply it to our bodies and hopefully translate that into people's effectively changing people's health in their lifetime, I think is pretty cool.
That's such a good question. You could get an entire PhD just looking at that. So translating that to diatoms are super, super old. They've been around for 100 million years. They predate multicellular organisms. They've been doing this for a very, very long time. But on the flip side, these micro scale, really quick interactions can completely rewrite their physiology in just 30 minutes. So you are taking these critters that have been doing the exact same thing for 100 million years, and just by changing the organisms that they are effectively swimming next to, you can completely rewrite everything that they're doing. And the exact same thing is true with us. I mean, talk to someone who has had like ulcers caused by Helicobacter pylori. You change their microbiome and they're cured. And it just, this incredible power of the microbiome that I think is very astounding. Super cool. Super cool. Yeah, the amount of evolutionary distance between two species of diatom that look effectively exactly the same is as much as between an elephant and a human. So if that gives you a sense of how ancient these organisms are, and this is just one species that they looked at in this study. There are hundreds of thousands of different species in the ocean, and how those will be affected by climate change, how that's changing the relationships. I guess that's also probably a topic for a different Planet Microbes. But I have some study questions. How many people do we have now? Should we split into groups, do you think, Jasmine, or stay as a cadre? Probably we should split into maybe 2 different groups.
Yeah, we have about 18 people
Oh,
total,
perfect.
including you and I. So yeah, we can split up into a couple of groups if you like.
And I can share the Google, oh, perfect. There it is. So Jasmine just read my mind and shared the Google Doc that has the questions. If you want to jot some answers down or questions that you have, feel free to skip around or the best part about Planet Microbes is when these discussions spiral off into totally random places. So feel free to take these just as a springboard and talk about whatever you want. And then we'll come back together as a group and have a final discussion. And I will also pop from one room to the other over the course so that we can all have a smaller chat.